Great. All right, thanks, Mackenzie. So I'm going to start and I'll give a quick overview of what we're going to talk about today. Then we'll switch to Chase, and I will come back to me. So thanks everyone for coming. Hopefully, we can share the topics Chase and I are going to discuss today. I think are some of them two topics we're the most excited about that's coming to um, the CMDKP and also all of our other knowledge portals. Some of them you've heard of, but I think they're starting to mature to the point that we have, I, I think, some exciting data for people to see. So, the title is Exploring Mechanisms Underlying the Human Phenome with PIGEAN and CFD Reveal. For those who don't know me, I'm an assistant professor at Boston Children's in the Broad, and I lead the technical and scientific development of the portals. We also have a lab that does a lot of inference of genetic support and human disease mechanisms, and Chase is a postdoc in the lab who's going to be talking about his main project. So, for those who have been attendees at the webinars, these topics we have covered before. So just to give you a quick background, we will go into more detail on this, but um, the first topic we're going to talk about today is a method called PIGEAN that we first talked about a year and a couple months ago now, which for those who use the human genetic evidence scores on the gene page of our knowledge portal, someone I think is unmuted, just as an FYI.
Okay. Thank you, everyone.
So so the huge scores, this is a paper that we published in 2022. That's a very simple way of identifying whether, the strength of evidence between a gene and a trait. And this method called PIGEAN is an extension of that and something that, is still unpublished, but, nearing publication. And we'll soon be replacing these huge scores with something I think Mark called huger scores, human genetic evidence refined, I think we're going to call it. But also introducing this additional concept of indirect genetic support that really makes a huge difference in terms of the genes that you're able to implicate in a trait. And the basic idea is if direct support tells you, is there a GWAS hit nearby my gene? Or is there an exome signal for my gene? Indirect evidence evaluates whether there's genetic support for a pathway that the gene is involved in. And the way that's done is through a method that catalogs a ton of different genetic annotations or gene sets that kind of track the pathways that the gene is involved in, and then uses human genetics to find which of those pathways are relevant to a trait and then prioritizes genes within this. And this kind of Bayesian approach that I think Chase will review briefly, but really we'll go into more detail on why we think this is quite an advance beyond what's on the portal today. And then I'm going to switch and we're going to talk about how this actually has uses well beyond just genetic support between a gene and a trait, but can start to really allow you to probe mechanisms beyond just the single gene level. And we're going to talk about this in the context of the common fund data ecosystem. So while I think this was a CFDE webinar, back in January of this year, we talked about how we are applying PIGEAN to extract mechanisms that are relevant to traits within the CFDE so that people who are interested in diseases can find things in the CFDE that are relevant to those. And we talked through a basic approach to doing this back in January. And producing a huge catalog of these mechanism to phenotype relationships. But we now have a new method that allows much, much more flexible and interactive query of these via like an AI workflow that we think is like a really powerful idea. And this is now available on our CFD site. So I'm going to take you through that after Chase, and hopefully something that you all can start playing around with immediately. So that's kind of the roadmap today. Chase is going to talk about PIGEAN and in particular validating why we think the data is quite good for that. I think we're going to focus a little bit on the PIGEAN portal, which is already available. It's not linked from our site. We'll try to keep that to about 20-ish minutes, Chase, right? And then we'll switch to how that's being deployed into the CFDE. If there is, like as Mackenzie said, since this is a focus group, there's kind of a blur between these and the webinars, but we do make these a little bit more interactive. So if people have questions, please put those in the chat and we can, and if we end up taking more time on the first half, that's fine. We can abbreviate or shorten the second half as well. Okay, so with that, Chase, I'm going to turn it over to you to share, and you can take us through the updates on PIGEAN.
Sure, thank you very much. Let me see if this will just work. Can you guys see my screen so far?
Yeah,
Yes.
thanks, Chase.
Okay, great. So I probably made too many slides. We don't have to get through them all. I can rush through them. It'll be fine. So let's start. Let me switch this. Okay, do you guys see this?
Yes.
Great. So, like Jason said, I'm a postdoc in his lab. I'm going to be talking about PIGEAN here. He gave a little bit of intuition. I'm hopefully going to maybe give a little bit more. I'm not going to try to not go through too much like math, but just give some like math adjacent intuition, just because I guess you guys have seen this before last year, but maybe just as a reminder. It also helps build the story, I think, in terms of using the resource. So hopefully that comes through. So, the basic idea of PIGEAN is we want to figure out how to find genes relevant to disease. So, when we run a GWAS, which is this sort of example GWAS on a gene level here, you might have disease-associated genes that are implicated by GWAS. So, these rise above some kind of significance threshold, but we know that GWAS doesn't capture everything. So, how do we find these missed disease relevant genes that somehow arbitrarily fall below this threshold? And the idea of PIGEAN is, can we boost genes in these shared mechanisms or in these shared gene annotations of gene sets? And so the basic idea is, let's say we have a set of genes that are all implicated together. This could be pathways, this could be other things like string DB that are linking like RNA-seq signals or whatever it is, doesn't really matter. But let's take the mechanism as our main like theoretical underpinning here. So maybe we have two gene sets and they're involved here in these two red dots, but nothing else that is really relevant is involved in this. But then we have like another gene set, and it involves both of those two disease-associated genes that we capture from GWAS, and also that missed disease-relevant gene. And so what PIGEAN is trying to do is let's try to boost these signals. So let's use these other disease-associated genes and shared pathways to boost signal of potentially missed disease-relevant genes. And as we do this over and over, and we have more and more evidence that suggests these two genes are linked, then maybe we can boost that disease-relevant gene now over whatever our significance threshold is and potentially find all relevant genes. So we're always making the assumption that genes associated in GWAS are always relevant because they show up in the population. But if there's a disease-relevant gene that's missed because it doesn't show up in the population because it's more of a rare variant, but maybe we can still recover that by boosting signals from these pathways. And feel free to stop me at any point if like there's something that's unclear, but this is the basic intuition. Okay, so that's what GWAS would find, right? And then this is what PIGEAN would find. So doing this. Started from here, now we're here, right? Okay. So a little bit about what PIGEAN actually is, and this is my little nice, hopefully it's funny, Flight of the Valkyries, right? Flight of the Pigeons, hopefully. We're a big bird group, so let's talk about a little bit how this works. So PIGEAN is basically trying to fuse a couple of different data sources together probabilistically. So we're starting here on the left with prioritization via genetic evidence. So this could be burden tests, GWAS mainly, or what we've also found to be really successful is just mining these Mendelian disease databases like Orphanet. And since everything's a probability in our framework, we can very easily port that over into an interpretation that we can run PIGEAN on. Okay, so we have this like direct genetic evidence here on the left. And then there exists all these gene annotation databases. So Reactome, right? All these different RNA-seq enrichments from GSEA or whatever you're doing, laboratory experiments that maybe you're running behind the scenes. And a lot of gene prioritization is done with these other resources. And the prioritization that you get from both of these might not actually match, right? What I'm getting from genetic evidence and what I'm getting from these other more holistic heuristic databases, they're not necessarily going to jive. And so the question is, can we integrate these in some kind of robust manner? And the way that we do this is through uh, basically a logistic regression problem. So we have some direct support. These are going to be our huge scores in the portal. These are giving us our genetic evidence. And for our indirect support, we're going to basically build a gene annotation matrix that is going to basically link all of the genes in these shared pathways. So we can think of this matrix as ones or zeros, and each of the rows as a different gene set, each of the columns as a gene, and you put a one in it if it's in the gene set, and you put a zero otherwise, or you could even just take a continuous approach and you could actually have like some kind of robustness as a metric here. So it doesn't have to be just one or zero, but that's the easiest interpretation. And then what we want to do is figure out what the effect sizes are. So this beta in the center, what these effect sizes are of each of these gene sets. And that will give us what our interpretation is of this indirect support quantity of a gene. Because what's going to happen, so the other thing is, so what we're capturing from these indirect support, or where we're sort of mining these from, and Jason touched on this, is like things like MSigDB, right? Mouse phenotypes, string, a bunch of other stuff, right? So that's all what's populating and building that gene annotation matrix. And please feel free to stop me if this is confusing or fast, okay? And so then the question is, how do you calculate what these betas are? And then how do you actually calculate what is disease, which genes are actually disease relevant through this framework? And this is where a graphical model plays a really nice role. So this is basically what Jason showed, just kind of shifted over a little bit. So we have some direct genetic support from our huge scores. We have some latent variable that's going to be a one or a zero. We're trying to calculate the probability that this variable is a one, meaning the gene is relevant, or zero, meaning it's not. And the way we're going to calculate that is by fitting this model towards the right-hand side. So all of these betas get instantiated. You have observed genetic evidence coming from huge scores of all the other genes in the genome for this trait. And you're basically wrapping this probabilistic graphical model in a Gibbs loop, and you're constantly solving this logistic regression problem over and over until you get convergence on both the betas and the relevance latent variables. So that's essentially what's happening here. And once you do that, it opens up this really cool continuum of genetic support across a ton of traits, because this can actually run really, really quickly. And so that's what you're seeing over here on the left. So naturally, we have a lot more associations than what you're just seeing on the screen. I just randomly sampled from this huge decomposition. So we have about 7,000 phenotypes. Now we can put them on this continuous axis of direct support coming from our huge scores, our indirect support coming from this fitting that you're seeing on the right, and figure out a bunch of interesting things. So to sort of break this chart down, and I'll kind of show this in the portal too, how this all sort of breaks down, is we can look at different sections of this graph to tell us different things. So this section over here, can everybody see my cursor? Can I get a thumbs up if people
Yeah,
can
we can see it, Chase.
Great, thank you. So over here on the upper left-hand corner, these are going to be genes that have really high genetic evidence. So like really high GWAS associations or really high exome burden tests type of stuff. But they're not well studied. They don't exist in a lot of pathway databases, especially the things over here. That means like there's no actual indirect genetic support for this. So no, it doesn't exist in any of the biological knowledge bases, at least that we were using to fit this model. So this is interesting, right? So these are like novel genetic associations for a trait. If we come over down to here on the purple, these are genes that are well studied. So they're in a ton of pathways, but there's not much direct genetic evidence for it. So this is another axis that might be interesting based off of the analysis that you want to do. And naturally up here, these are well-studied genes that also have high genetic evidence. and you tend to see things that are like very well known, right? Insulin and type 2 diabetes, that's like the top one here, right? AP, APO with total cholesterol measurement, right? Very, very well-validated, indicated genes. So when we saw this like a year ago, we're like, that's pretty cool. Like that seems to be making sense. But what we also need to remember is like this is really a continuum. So unlike when you're looking at GWAS associations and you're picking like, a p-value threshold and then everything above that gets associated, we don't really have to do this because this is a fully Bayesian probabilistic model. So there's really actually interesting things even lower down on this continuum. So a ton of things to mine that we think you guys will find pretty interesting. So just to give a little bit of breakdown of what this whole resource is. So the total traits, and I'm sorry, I haven't calculated how many HPO terms there are. Maybe Jason could fill this in. But the model that I generated this from, we have 5,535 traits plus all the HPO terms, which gets you above 7,000 phenotypes. The number of traits we have from the GWAS catalog is about 3,000. From meta-analysis from our A2F Association of Function portal, we have about 1,000. And then we've also ran this on a bunch of orphanet rare diseases, so 1,523 rare diseases, which gives you a whole landscape of like 110 million plus associations to mine from and to study. Okay. So that's sort of the PIGEAN recap. How am I doing on time? Okay.
Yeah, you're okay.
Yeah, you're good.
Okay, wonderful. Any questions so far?
I don't think so.
Great. So let's talk about if we can actually trust this resource, which when Jason brought me onto this project, that this has sort of been my job for the last year. And hopefully what I'm going to convince you of is we have this huge landscape and you can trust it. So that's what I'm going to try to prove to you. So we already, I think, with this webinar audience, we already trust the huge scores, right? Those are already on the portal. We're already using them. So that's great. So now what I'm going to focus our attention on is can we actually trust this indirect genetic support? Does this actually hold up as well as like the huge scores have held up? So the first test that we wanted to do was, can this indirect support axes predict direct support on held out chromosomes? So we chose hopefully a representative set of about 14 to 15 traits that had reasonable power. And we wanted to test this. So basically, we ran this cross-validation. We held out the genes on all the chromosomes, ran PIGEAN on the non-held out portion, and then regressed that indirect support for each of those genes on the direct support calculated by huge on that held out chromosome, and then looked to see if we're actually predicting this. And in all cases, we have a pretty good signal here. So it seems that indirect support is predicting direct genetic associations on held out chromosomes, as we would hopefully expect to see, which is great. So check. We've done that. The next thing we wanted to do was understand for our rare disease case, can we do a similar analysis? So for these rare diseases, the way we calculate PIGEAN is we take a ground truth disease gene list from orphanet or from OMIM, and then we basically run PIGEAN on that set of genes, treating those genes that are in OMIM or orphanet as like a high probability of direct support and everything else setting to a background and then running until convergence and seeing what happens. And so what this analysis is going to show is I ran this over, I think it was 40 orphanet traits, both of groups of disorders and individual disorders. And I ran this as a cross-validation experiment. So I held out a ground truth gene each time. Then I wanted to see how well PIGEAN would bubble up that held out gene using only indirect support and capture it again. And so this is the ROC curve that you're seeing for this. So as you go up in the top Ks predicted by indirect support, how often is that held out gene in that top K? So we can see we go very quickly from 0 to like, I think this is probably like 100 to about a point 75% recovery rate, which I think is pretty good. And as you keep increasing K, so this is the top genes that you're looking at, we basically converge to like 90%, which I think gives me a lot of confidence that even in the rare disease case, we seem to be recovering ground truth rare disease genes. Okay, so shown cross-validation on a chromosome level, showed it on a rare disease level, check. Now, what we wanted to do is, that's great, but your model is using gene sets that might be circular. This is the big critique that we've always gotten with PIGEAN, is that how are you handling circularity? If gene sets are coming from sources that are implicating GWAS already, maybe you're just double counting in some kind of way. And so what we wanted to do was control for this and ask this question of, does indirect support predict future GWAS signals? So the way we did this is we identified from our portal, we identified two GWASs per trait, one that was pre-2018 and one that was post-2018. And where with the additional criteria that the earlier study had lower power. And we found on our portal 67 traits met this criteria. We then ran a magma on both the pre and the post 2018 GWASs to calculate z-scores. And we're using this as sort of an independent validation. I think huge is better than magma, and I think we've shown this many, many times, but we didn't want to be circular in that analysis either. So we were using magma to calculate these z-scores and use that as our proxy for calculating a GWAS signal. Then what we did is we ran PIGEAN on the pre-2018 GWAS using only gene sets published before 2018. So there's no way gene set circularity could have happened in predicting that future signal. Then what we did is we regressed that pre-2018 magma score on the post-2018 magma score to get our baseline R squared, to get our baseline fit, right? To see how well did past magma scores predict future, right? Then what we did is we took PIGEAN and our indirect scores to filter genes that PIGEAN has predicted will be future enriched for a signal. Then we reran that regression on those filter gene sets, again, magma on magma to not be biased, and we computed an improvement in R squared. So that was our study design here, and what you can see, this is sort of the result of that. So as you increase your indirect support threshold, I'm sorry the numbers on the bottom are a little small, but you're increasing from basically 0 to 1, or 0 to 4, and this is in log odds. You're increasing your R squared improvement, so your fit on that future GWAS signal in a statistically significant way. And it's like going from basically when you don't do any filtering, you're at like basically 0. And when you increase that, I think you're coming at about like 0.4 increase in like R squared fit. So that's pretty cool. So to me, what that's saying is indirect support is being predictive in some capacity of that future GWAS signal. So check again. Great. So these are all like very, I would say, genetic-based validations. So the other validation we wanted to do is try to validate this on some kind of independent axis that wasn't necessarily genetics, and see if this prioritizing using PIGEAN could actually support some other type of use case. So there's been a couple of papers that have basically shown that human genetics evidence can increase drug target success. So 66% of drugs approved in 2021 had some genetic support. And this has been shown again and again. 63% approved in 2012 to 2022 had genetic support. And this was proved most recently by Minical et al., where they looked at how much genetic support increases your likelihood of succeeding clinically. And what they found in that paper was if you have some kind of GWAS association or some kind of genetic support, you can increase the likelihood of a drug program succeeding by 2.6 times. So that was a pretty cool finding. So naturally, we want to ask this question of, can PIGEAN predict drug targets? Because this would give us a nice independent access to discuss utility and validate PIGEAN. So the first thing we did is we reran the study that I just showed you before. We ran this on PIGEAN. What I'd like to point you to is up here. This is only indirect, and this is broken down by the source categories of where we're getting our data from. So down here is open target genetics. We're seeing like sitting around 1.9, right? These other sources give you a little bit higher, and that 2.6 number that Minical cited is a combination of basically all of these sources, right? Because you can see the OMIM is much more likely up here, so that's pulling things up. But the common diseases that were found here were sitting at like 1.92, right? So naturally, we can see PIGEAN in terms of like our combined support, which is this one here. We're going like I would like to look at like this combined clinical category of both historical and active clinical programs. Like now we're sitting at like a 2.5 area here in terms of a fold increase. And similarly, indirect support is higher than the direct support from all of Open Targets, which I think is pretty compelling. So the question I think, though, you should immediately ask that is to challenge this is, well, what about if your gene sets that you're using to fit PIGEAN were coming from drug target studies or were coming from papers that were talking about drugs, which is a very natural question to ask. So the next thing we did is we did a temporal holdout. And so that model that I just showed you, that pre-2018 model that we ran, we did this again, and then we held out all of the drug targets that were post 2018 and only predicted on those. So we ran this pre 2018 PIGEAN model where it's not even possible that we could have even had those potential drug confounders in our model, and then only predicted on future drug targets. So we did this, and I'm showing you both the direct, so these are the huge scores, and the indirect as a function of increasing your limit of direct or indirect support. So getting more specific. And what you'll see in both of these is that like we nicely, for direct support, we nicely come above like the magma support, magma prioritization line here. And we're like getting it to like 3.5 here, which is pretty crazy. And then this other line here, we're just showing the number of total targets that's predicted as a function of that limit. So we're starting with like 1,200 targets over here, and we're reducing down to what, less than 100 targets as we're sort of coming down, which is natural as this method gets more specific. Down here is indirect as well, and we can actually see a nice little build across this, centering around 2. It looks 2.5 or something up here, and then coming down again as you like lower the number of targets. But the other interesting thing is if you notice these two lines here, we have a ton more targets for indirect support than we do for direct. So like that direct kind of has this really sharp decrease, and indirect kind of stays slightly more flat, I guess. And we're getting similar relative success for both of these independently, which is, I think, very, very nice. So maybe check, right? Like I don't think there's any way a future drug program could have influenced this analysis. So it seems that PIGEAN can predict future drugs that are clinically likely to succeed. Then the question is, how many more targets does PIGEAN actually give you to sort of prioritize over and study? So the light regions here are the completely unique targets. So these are unique to any other source that you're seeing here. And then the total targets is just, this can be combined with other things. So naturally, PIGEAN gives you way more targets. And this is only on one data set of this pharma projects data set that was used in this other paper. But it doesn't have to be limited to this. Like this is a lower bound naturally on likely the relevant targets that are likely to clinically succeed, which is the case that's made in other papers as well. So more targets, similar or better relative success, that's great. The last thing I want to kind of touch on is recall that this is a continuum, right? So what I was sort of showing you here is I'm kind of thresholding this at discrete values. But how do I know, like, really, I'm just doing this because that's to compare fairly against all the other sources. That's what you have to do. Because if you just have an association in a database without a score attached to it, you kind of have to have some sort of calibration in terms of just to be fair. Okay. But really, this is a continuum. So there's probably going to be interesting things that are below these thresholds, especially for different traits. We'll have different thresholds for these kinds of things. And the only other real resource that provides sort of a continuous association score is Open Targets platform, which I'm sure you guys have, it's pretty famous. I'm sure you guys have heard of it. And they kind of have a version of what I would call direct support and indirect support. So they have direct support from GWAS associations over on the left, from ClinVar and ClinGen and Orphanet and all these things. And then they also have this other, what I would call like an indirect support light uh, type of association. So this Europe PCM, this is mined literature, so a Jacquard index of the association between a target and a phenotype. Mouse phenotypes here for IMPC. So they have some kind of notion of this, right? And what they use to sort of combine all these things is just a harmonic average, which is to me a little bit uncompelling in terms of just how you fuse these data sources. But one thing that we can do is we can actually compare now to open targets. We can compare our continuum to theirs in terms of relative success. So we asked this question. So PIGEAN is the dashed lines that are here above, and open targets are what's here below. The combined support of open targets is this value here, so their total association score. So this thing. So that's what's sitting here, which is quite it's quite rough. And so I'm sorry, you can't quite see this on the bottom. The x-axis here are basically if you sort your continuum in a descending manner, these are like the top portion of the target indication pairs that your method is selecting. So as I go from left to right, I'm including more and more and more potential targets that are supported by each of these methods. Relative success is on the y-axis. So I'm sorry that this is small, but this is 1 and 2 and 3. And we can see that PIGEAN like really outperforms for like a majority of the top percentage of the associations in our platform. So like open targets is only really successful like at that where that other paper said of like 1.9, that's like only about what 1%, the top 1% of their associations, where like PIGEAN is sort of maintaining that same level of success rate in our top like 25% of associations. So I think this is quite compelling in terms of comparing this continuum across this relative success category. So I would say PIGEAN is giving you something that open targets can't really give you. So check again. So just to recap, and then we can maybe go into a demo, is it seems like indirect support predicts direct associations on held out chromosomes for common traits. We recover rare disease genes, so we recover some ground truth there. We can predict future GWAS associations. We have a maximum relative success rate when combining both pieces of evidence of about 2.98. Indirect alone, the max was 2.6, stabilizing at about 2.4. And even when you temporally hold out future drug targets, we can predict on that too. So now I just want to kind of put this picture back on the viewport here and first ask if there's any questions, and then maybe we can use this to start sort of our navigation of the portal and see what you guys think.
If you have any questions, please feel free to raise your hand and come off mute. And you can also use the chat.
I see some comments in the chat, but I don't think any questions so far.
I have a question.
Yeah.
So yeah, I work on mouse genetics, and there are a number of genes where there's incredibly strong evidence from knockout models and from mutations in mice. And then you go to the human GWAS, and it's ice cold. And I just wonder if you have taken on any of those and run it through your algorithm to see if you can find support when you combine all these data sources.
Yes, so I will take that. So as we segue into the demo of the portal, what you will see is for most GWAS traits, there are mouse phenotypes that are strongly enriched for them and that predict the GWAS. So one of the things Chase is focusing here on the gene to trait linkage, but you also get the gene annotations that predict genetic support. And the mouse model is the single best predictor that we have found. And there are highly predictive. So there are some that are like almost funny how much of a match the mouse phenotype is to the human genetics. Now, as you're saying, that does not mean that every gene that has that mouse phenotype has a direct support, but you do now pick those up from the indirect support. What's also interesting is that there are many mouse phenotypes that semantically match the human phenotype that you do not see indirect support. And PIGEAN kind of like filters through those mouse phenotypes and combinations of them that are actually enriched for the genetic trait. And we actually have a resource on the Common Fund that systematically applied this. And there's a whole module where you can type in a mouse phenotype and see the human traits, and conversely type in a human trait and see the mouse. And we could go into much more detail about this, but I don't know if that's what you're asking. But basically, what I've been amazed is how much mouse signal there is, relative to some of these other libraries people use. Now, one other just final point I'll say as Chase pulls this up is just that holds for some trait categories more so than others. So the metabolic traits, the mice are incredibly good predictors. For things like the mental disorder traits, you see things like autism or schizophrenia are enriched for like these abnormal exploratory behavior phenotypes, which makes sense. But the mouse are weaker predictors for some trait categories. And that's something we're exploring and would be happy to talk further with you about if you're interested in being involved in that.
I would love to talk to you about it, Jason. But let me just make sure I understood you here. These were instances where the ones that you were just telling me, where the mouse was an excellent predictor, were those instances where just the straight up bare GWAS didn't show anything? It was the combined analysis that did?
So let's look at an example. Actually, Chase, can
Yeah.
you type in my favorite is like chronotype morningness. Because this is not one I think that you would necessarily. Okay, so chronotype morningness, that one right there.
Yep.
So if you look down at, so we're kind of jumping ahead here, but if you go to the second card below, these are the top ranked gene sets. One more card down. Okay, so you can see that like the top one is like MP shortened circadian behavior period. So that's the single best predictor for predicting a GWAS association, right? Now, if you expand the genes tab on the right, what you'll see is this continuum that Chase had, right? So all these top genes, the direct support column indicates whether there's a GWAS hit. So the top ones here all are like have mostly very strong GWAS hits, right? But if you start paging towards the next tabs, you'll see an example like here is NR1D1. That's your example of like an ice cold GWAS hit. But because the mouse phenotype is such a strong predictor, it gets pulled up by the indirect support to have like an overall 90% combined probability because it's so strong, this mouse phenotype. So this is one that's picked up by the combined support. So it's like there's lots of reasons, in other words, why you wouldn't see a genetic signal, right? One reason is like there's just evolution, maybe the gene's constrained for many reasons. So the whole pitch here is that not all disease relevant genes will be disease associated. And this is one example where we would predict that it would be, right? There are other mouse phenotypes, though, that they're not as strong predictive. So just because it might look like it's a strong one, you might not see that. So that's kind of what we're trying to model. And like as Chase is saying, there's 110 million of these relationships, which is why we're trying to share this and get the word out, because we think there's a lot of value in people like you browsing this to like really dig into it.
Are you gonna continue on and really walk us through how to do it? I really don't know how to read this table.
Yeah, so like I think actually we'll just spend the rest of, so we'll kind of skip the reveal stuff today and like we'll just go through this portal in more detail. And Chase, you want to take us through it? I can also jump in as you go through. So maybe
Yeah,
you
feel
start from
free.
the beginning. Do you want to start
Yeah.
from the beginning?
Sure. Yeah, feel free to jump in at any time, or if you think I'm pulling focus somewhere, just
All right, you got to go. Yeah, the PIGEAN. There's no. So for those, the link here is you put /PIGEAN after any of our portals, and you will get the same data. Okay, we're demoing a dev site here, which has more phenotypes, but the URL's up there. I'll put it in the chat to a2fkp.org/PIGEAN.
Thanks,
And
Jason.
yeah, and so like I said, there's not linked yet. One of the reasons we're sharing this is so people can start to use it and give us feedback as we start to integrate it into the main portals. But this has all of the data that Chase shared, and he'll walk you through how we use it.
Okay, great. So first, like, well, I'll just ask the audience, does anybody have an interesting phenotype that they're like interested in particular? If not, I'm just going to pick one.
Hypertension.
Hypertension. Okay, let's do it. So we type this in, right? So tension, and we'll see there's like a ton of different options here, right? So it's still, I think, kind of an open question of like which GWAS do you pick for hypertension, right? But let's pick one that like I know will sort of work. And you can kind of see we have descriptions of these. So I think they mainly make sense, right? But there's a lot of different types of hypertension GWAS with background traits and other things, right? But I think I would pick this one. Yes. So if we walk through hypertension, the first screen we're going to hit here is kind of that continuum that I showed you. Remember that like direct support by indirect support. But now instead of looking at that whole cross-trait landscape that I had shown you with a bunch of other points, these are only the results for hypertension. And remember, we have our different categories. So up here, we have, okay, this gene FAM109A. You're going to have to be gentle with me. I'm not a geneticist, so I'm not going to know what these genes do by trait, but I can copy to Google or ChatGPT and I can find out what they do. So over here, we've got all of our associations that don't have any indirect support, so they're not in a shared gene set, but they have a GWAS hit, right? We got a GWAS hit over here. Great. And these are novel associations because the other way you can view like if something is in a gene set, right, has this gene been published in a paper somewhere? Has somebody curated it? Has somebody studied it in any kind of way that would lead to an eventual association in some other kind of heuristic resource? And if they haven't, this is sort of novel. So up here are your novel GWAS hits for hypertension. And I don't know if somebody wants to Google or chat GPT, well, like one of these genes with hypertension, maybe Jason, we could probably show you like maybe that there's some evidence for something up here. Great. On the other end of the spectrum over here, right?
Actually, when you get to the table, I'll show you how to pull the GWAS evidence. Yeah.
Okay, cool. So when you get to this side of the table, right, we got the no GWAS hit, but it's in some kind of shared gene set. So that's what HEY2 and VEGF-A are, and these different other genes here. So, and then up here we have our well-studied and GWAS hits, right? So this is ACE, NOS3. Is this a hypertension gene? I feel like this should probably be a hypertension gene. I hope it is.
Obviously are like yeah. So those are almost always your very, very well known genes.
So that's good, right? So now when we scroll down to the bottom here, we can get a table view of this. So this is on the gene level. So now we have like it's always going to be supported by combined support here. So this is both the indirect evidence and the direct evidence. So it's going to be.
And one point, just to jump in, if you scroll down. So the units are this combined probability is a probability. The other quantities are all in log odds. So just in people aren't familiar with that, you can convert any probability to an odds, right? So 3-to-1 odds, if like you're a bettor, you'll use that all the time, is like 75%, right? And then you take the log of that. So that's why all these are in units of probabilities. They're just log odds of that. Okay.
Yeah. Yeah. Great. So we have this table, and now we can show gene sets. And what this mechanism is, Jason, this is still generated by LLM, right? Based off of the
let's not. If we have time, we'll get to mechanism, but let's just focus. Let's ignore that column for today and just focus on the genes, I think, and gene sets. Yeah.
So for this gene, for NOS3, these are the top gene sets that are implicated for that prediction of this being likely to be disease relevant to hypertension. Okay. So decreased heart rate in mouse, abnormal heart left ventricle morphology in mouse, increased heart weight in mouse, regulation of blood pressure. All of these, when we're looking at them, seem to make sense, right? I'm sorry if they're a little bit complicated to read with the underscores right now. Um,
Yeah, we're working on fixing these, but essentially the MP is anything from the MGI mouse database. And then the others are from MSigDB, if those are familiar with it. So GoBP is like the gene ontology. So you can, if you type in any of these terms into Google, it will define it for you. And we're working on fixing that.
yeah. So pretty cool, right? So now I know where my indirect support came from, right? This is the provenance of that information for this gene. And naturally we can do that and we can sort of tab through all of these things. So I would say if something is like starting to be below 0.001, then like we're starting to get a little bit of noise, but we have really good regularization here. So this generally what this joint effect size does up here is it's usually pretty clean in terms of parsing the right things on the top couple of pages here.
And then pop open the view locus plot. That's where you can get the actual genetic direct support. So this takes a little bit of time to load, but will give you a view of the GWAS hit nearby. So this is the full dissection of where these two axes are coming from. What Chase just showed is that x-axis, right? Exactly what annotations were calibrated. Remember, these are all learned. We're not feeding anything into the model that says like these are what you should use for hypertension. We're giving it a giant library. It's learning from the full GWAS what's enriched, and then that's the provenance there. And then this is the provenance that tells you why it prioritized the direct support. That will be much stronger. You're always going to find some type of GWAS peak nearby it.
Great. Questions so far for this panel?
Just a quick question here.
Sure.
So you began with a phenotype-based
Yep.
screen. Does it always have to start with phenotype? Can you start with a gene?
Absolutely. Let's do it. Do
So
you have
do
a
you
gene
have
you're interested in by chance?
Yeah, well, I can give you the name of the gene that we work on that is ice cold in humans, but has a very strong phenotype in the mouse. The
Let's do
human
it.
is called ZNF148.
So we'll load this up. The first box we're going to see is a PheWAS plot of this. So we're going to see which traits has this gene been enriched with combined genetic support. So both GWAS and with our indirect support quantity that we mentioned. Okay. So that's what this chart's telling you here. Things that are above this line are going to be like very enriched, right? So vitamin D levels, looks like vitamin D measurement. What is this? What
waist hip
BMI of
ratio,
some kind?
waist hip ratio adjusted for BMI?
Waist hip ratio adjusted. Um, Not sure what that is.
Uramurate.
Something. Then, though, what's nice is so remember these are traits that will have some kind of combined support, but we have a little chart over here, right? That puts the PheWAS on that axis that I've been talking about. So on this axis, so these are the traits where this has a GWAS hit. Okay. And on this axis, this is where these are traits that have that gene implicated in indirect support. So cold GWAS hit traits that we can search for. So maybe we pick the top one here and we can go to it. Okay, and now let's see if we can find it by looking at
You can actually filter on if you type in the gene on the top.
right. ZNF148. So that's like here. Okay. And if we show the gene sets here, most of them are mouse related to,
The top one, which is the dominant contributor, is
yeah.
where Sesto responds to UVC5 is. If you go back, actually, though, Chase, to the gene
Yeah.
page, so just a comment, you can actually access this from the table here too. Because for each there, if you scroll down, oh, sorry, you have to go back one more to the gene actual page, the back button. Yeah. So if you scroll down, you can access the same things here. So for each of these traits, you can click on gene sets to expand. And you can also, for the traits that actually have full summary statistics in our portal, you can see the locus zoom plot. Those are going to be empty because they have no indirect support.
Oh, yeah.
But one point I would say to you, Alan, like if you scroll up, like typically the pattern on this right-hand plot is usually quite interesting. So what you're seeing here is a gene for which the genetics just does not match the reported biology in the literature because there's almost nothing along the diagonal, you know. The genes that are typically the genetics lines up with what's been reported are going to be have more things along the diagonal. So like just to give an example, like type in SLC38, which is an islet like zinc transporter. So
SLC 38.
38, yeah. Sorry, C30.
38.
Yeah, then an
A,
A.
A.
A8.
Yeah. So then
Like
hit
this?
enter. Yeah. Oh no. SLC here. SLC
Yeah.
30 30
Three.
30.
3-0.
No, no, you have a zero.
3-0.
A8.
A8.
8.
Got
Like
it.
click on that. Yeah. Make sure
Thank you.
you have to click on that and then click go. So what you'll see here is like an example of a gene that that there's many more things like kind of along the diagonal, and those are the things here are going to be the ones where you kind of have an explanation for the GWAS hits, right? So there are things on the left that it's associated with that the traits are not enriched for, and there are things on the bottom axis that are traits for which there's known biology, but you don't see the associations. But you start to have some insight into like how the known biology is capturing the association. So there's much more we could say about this. But your gene is an example of one where there really genetics is just not lining up with the biology for whatever reason that might be.
So is a major reason that genetics is only looking at SNP variation and its association, but the other sources of information you're pulling in have a lot to do, are often related to gene expression, right?
Yeah, yeah. I mean, so like the traits include the rare diseases too, right? So there, yeah, like I don't know the full reason. There's lots of explanations. We probably don't have time to get into that, but it's something we're actively looking into. It's like the patterns of this two axes for one gene across many, many traits gives you like a fingerprint that you can use to kind of, at a glance, interpret how the literature stacks and other data sources stack up to the genetics. And, again, we ran out of time for this today, but one of the things we're actively exploring is ways we can leverage that information to actually do more complex mechanistic or whatnot queries. Yeah, Julius. And I think like since we have 5 minutes left, we'll just stop for questions now. Yeah, Julius.
Yeah, thank you so much. Really cool to see this. The longer I look at this sort of these direct support versus indirect support plots, um, the more I keep wondering about how this works with strong direct support but low indirect support. Because my intuition is that there should be, if I have a strong direct support, that this should show up in some sort of gene set as well.
Yeah.
So this top left corner of these plots I find somewhat puzzling because I would always assume, if the gene is sort of directly implicated, it would also show up in the gene sets that we would then again make it show up in the indirect support. Or is
Right.
that someone
Yeah. So
mistaken?
there's three reasons it could be. One reason is that well, I guess there's really only two reasons. One reason is that the gene just doesn't have a ton of like, hasn't been studied before. There's like a large fraction of genes that like don't have any annotations in databases, right? And those will almost always show up as the vertical line, right? Now, what is more interesting to me are the cases like here, where like the gene is clearly well captured in databases because there are some traits for which it has indirect support. But like Chase, what's that upper left annotation? But for whatever reason, like, okay, so actually this is another second reason. This top one are for exome studies that do not have power to detect enrichment. So a lot of these traits that have low power are ones where you don't have enough from the genetics to find the gene sets that actually are enriched, right? So does that make sense? So like there's no, if you click, we're not going to click through this, but if you click through this, you would find no genes with like indirect support for this. So that is a large number of traits. But the third reason is even for there are examples where genes for very well-powered traits like have like high direct support, no indirect, and there are other, but it has indirect for other traits. I think that's a case where the gene biology is different from the main axes of biology underlying the trait, right? So, like for type 2 diabetes, maybe your main axes are 10 mechanisms, but then there's this one gene that's in a mechanism that is not that aren't many other genes share for this trait. So those could be arguably the most interesting ones, right? The ones where the gene has a known mechanism, you pick up another traits, the trait has enough power to pick up mechanisms, but the gene's still an outlier. Those could be like your true novel insights. And those are all things that you can just trivially filter through this. Again, and something that we're hoping to unveil with this paper is kind of like enabling resources where you can do those type of queries.
That makes sense. Yes. Thank you.
Yeah, but it's something we're actively looking at. All right. I think we have, if does anyone have another one question or we're running low on time, but is it possible to
Good question.
predict FCC38 therapeutic direction? Not with the current. So as we described, the model here is strictly a binary, is the gene disease relevant? But as Chase mentioned, we have a bird team in the lab, and we have a method called Magpie, which we might be able to share soon, that does actually infer direction of effect just from GWAS data. Um, and we are happy. Well, hopefully we'll be sharing that soon and releasing that on the portal in the coming probably 6 months, I think would be the timeline for that. You could predict it from the exomes data that we have in the portal here, but that's not something that PIGEAN models at the moment. It just models a binary yes, no.
Jason, can you quickly recap how you get from the landing page of the PIGEAN portal to that graph that you've been showing us?
Yeah, so if you go back to /PIGEAN, if you just type in, this will accept a gene or a phenotype query. So if you type in the gene,
Yeah.
make sure you click on like the gene name. You can't just hit enter. So you have to click on search gene. But
Okay.
that will take you to the gene page. If you type in a phenotype, it will take you to the phenotype page.
But then that graph, yes. Oh, so that gives you the graph.
That will just open up. There's only two pages, basically. There's a gene page and there's a phenotype page. And then you can click from the gene page on a phenotype to go to the phenotype page and vice versa. But that's really all there is to navigating it.
Yeah,
And
I
my
don't
yeah.
that graph when I do that. Maybe you and I could spend some time on this together sometime.
Yeah, I'd be more than happy. Shoot me an email. I mean, like it might just not be loading, but it'll load. It should just load immediately, or not immediately, but it might take a couple seconds, but it should eventually load.
No, I have a bunch of things I want to talk. We haven't talked in a long time, so do you have time tomorrow?
Yeah, yeah. Shoot me
Okay.
an email.
All right.
Awesome. All right, I think that's it. So one thing I will just conclude, one of the, to tease this, and maybe Mackenzie, I don't know if we have an upcoming webinar. But one of the additional things this allows you to do is there's this higher order structure that all the genes and the gene sets provide that we haven't even talked about. But it distills the information in GWAS into these higher order mechanisms that we were getting into, which are tailor-made for mechanism first queries. So like, can you find me things that are traits that are associated with adipose expansion? You would have to manually filter through every trait, every gene, but a large language model can automatically do that for you. And that's what CFDE Reveal does. So if you want to explore on your own, cfdeknowledge.org is our CFDE portal, but maybe we will share that next time about how we're enabling these to do much, much more advanced queries than just gene and phenotype as well. And that'll be hopefully get your interest for the next webinar or one upcoming soon.
Yeah.
All right.
Thank you so much
Everyone.
to both Jason and Chase. Our next webinar is December 11th. And Jason, I'll talk to you about scheduling a follow-up to this because, like you said, we do want to talk about Reveal sometime soon. So if anyone has any other questions or concerns, please feel free to shoot us an email. We really appreciate all your time. Thanks,
All
everyone.
right. Thank
Bye.
you.